Good afternoon, ladies and gents. My name is Mrs. Cellular. Today we're going to look at formal test preparation, term 4 for grade 12s, grade 11s. So I just want to say the grade 11s, I'm sure they can feel that they're close to grade 12. They're just thinking about what they're going to do next year. So this is the last exam. Let's put in everything. Let's get good marks so that you can start grade 12 with a good feeling. Okay. So here we are, formal test preparation, grade 11, term 4. The grade 11s, I just want to show you all, there's choices to be made here for the teachers, all depending on the work that they have done this term. So the final assessment can be 2 papers, paper 1 and paper 2, like we are used to, before COVID. That is what we did, 2.5 hours, 150 marks, some of the topics in paper 1, some of the topics in paper 2. If for some reason your school could not complete the curriculum, or you are actually at school one day and the other day not, or the next week you are not there, so we are going to look at the test. The test for grade 11 will be 120 marks and it's going to be 2 hours. Something else I want you to take note of is that all the work that you have done for the whole year, term 1, term 2 and term 3 is going to count 60% of your final mark, and then term 4, the marks that you get there will be 40%. Okay, I say again, for those schools that could not finish the curriculum or that is actually rotating, this would be a good option. All right, so next thing, the types of questions you can expect. Just something else I want to make you aware of. You know multiple choice, you know about fit column A to column B, you know the way that your life sciences paper normally looks because you are in grade 11 already. Just remember, whenever you answer questions, look at the mark allocation, and according to the mark allocation, you answer your question. We need you to answer your questions in full sentences. Don't summarize your answer, because then we have to summarize your marks. Okay, here's a few examples of the type of questions or words, verbs that we can use. Okay, when you write your exam, use your highlighter and then you highlight the verb in the question. It helps you just to understand what do they want. For instance, here we have compare, where you have to compare 2 topics maybe, like what's the difference between photosynthesis and cellular respiration. They can even ask you to tabulate that. Then you have to draw a table and you will get a mark just for the table to be drawn. Then all the other information you put on the left and the right hand side. List very easy, name very easy. That is where you just give the basic answers. But the moment we get to explain, discuss, describe, then you have to make sure that you maybe remember that there's an A and a B section. You have to give the answer and explain something. For instance, why is the mitochondrion, or how is the mitochondrion or the lungs built to perform its function? Structure and function. So then there's an A and B section. So please be aware when it comes to that. Use your highlighter or your pencil or whatever weapon you choose in the exam, and you highlight or you underline the verbs and make sure you understand what to do. Okay? And then the format of the papers, just an explanation shortly again. If it's a paper 1 and paper 2 that you are writing, then it will be 150 mark papers. Paper short question, the normal short questions. You can have pictures and we ask questions about that. There can be a graph, or it can just be a straightforward question where you have sentences and you have to identify certain facts. The section B, there can be case studies, there can be graphs, there's a variety of things that we can use to test you, and there's a section, in section B, there's 2 questions, question 3 and 4. Now in this instance, that's why we have the arrow here for today. We don't have enough time to discuss the whole paper 1 and paper 2. We're going to discuss the test, and there you have it, section A, section B, the test for grade 11, Section A 40 marks, and then for section B there's 2 questions, 40 marks each. That will give you 120. All right. Next thing. We received this from our subject advisors to guide us on how to set these papers because we have to set it ourselves. So here you can see paper 1. These topics are relevant for paper 1 and that's the mark allocation for every section. Paper 2. These topics relevant for paper 2 and that's the mark allocation. And then for the one control test, if you are writing that one, there's the mark allocation, the different topics and 120 marks. Please be sure when you prepare for this test or exam that you know exactly what to study. And of course, ladies and gents, when we study, it would be very nice if you can have the ATPs with you. If your teacher haven't copied you these forms yet, please ask nicely. It helps you a lot to know exactly what you must study. For instance, this is now term 1, week 1. It gives you the microorganisms, viruses, bacteria, protista, fungi, what you need to know about them. Here they say one disease. The same with the bryophytes and pteridophytes, gymnosperms, angiosperms. You have to draw yourself a diagram with this, a summary about the vascular tissue, the true leaves and roots. Are we using seeds there or spores? Will there be a fruit, etc.? So you can use this to see exactly what is important. We have different textbooks, so sometimes you get confused. You don't know what you must now focus on and what you must leave. You must like to decrease the amount of work that you have to study. Okay, so please make sure, and this whole section, it's all according to the annual teaching plan. It helps you a lot. It's like your scope. All right, you must always want a scope. So let's start with the first section that's important here. Photosynthesis. It's an old topic that you do ever since the primary school. And when you look here on this paper, it says here, energy transformation to sustain life, photosynthesis. What do you need to know? What is the importance, the influence of different factors, and then just one investigation. So let's have a look here. Obviously, if you have science, you will also understand this equation. We start with the requirements on the left side. That would be water and carbon dioxide. Then light, obviously, chlorophyll that we find inside the chloroplast, and enzymes. They all assist in this situation. And then the product would be sugar, C6H12O6, and then oxygen, that is the byproduct. So plants use water and carbon dioxide with light, chlorophyll, and enzymes assisting to give us sugar and oxygen. Now the sugar is also for the plant, and obviously people use the oxygen that is now given off. All right, there we have just a reminder. Photosynthesis, all the different things that you need. This is the organelle, the different parts of the organelle. There's all the different labels. You must be able to label the chromosome, and then the function of every section. You must be able to tell us where is the dark phase happening, where is the light phase happening. These red blocks that I added here, that comes from support material that we received from the Aleph as well. You can find this on Mindstream. You can Google that and then you can get the support material. There is actually lots of information, summary regarding terminology, and then it's also mind maps that helps you to study. Okay, so get that for yourself when you study for this exam. Here they say the light and the dark phases, biochemical processes in which photosynthesis occur, NB mention only. So you just need to know that there's a light phase and a dark phase, and you just need to know where it is happening. And then the chloroplast, you must know the structure and then how it is built for its function. So just go back to the basics. Maybe you have forgotten them by now. All right. This is what they suggest. What is important about photosynthesis? The release of oxygen, uptake of carbon dioxide from the atmosphere, and then food production, obviously trapping the energy for the leaf. Plants are photosynthesizing so that they can have food, and then obviously they use the carbon dioxide. The next important thing, influence of the different factors. Now, light intensity, temperature, and carbon dioxide has an influence on photosynthesis. And sometimes we give you graphs, and you need to be able to understand the graph and then answer the questions regarding that. So please make sure that you know what is the influence of light, temperature, and CO2. There you can see they discuss that for you. Make a summary for yourself, and then you just need to do one investigation, and the one that was suggested on the ATPs would be the scientific process where light is necessary for photosynthesis. So just go and have a look at that one again. Make sure you will be able to identify the different parts of the equipment and all the equipment that you need to do this experiment. All right. The other section that is important, energy transformation, respiration. I go back to this one. Energy transformation, respiration. You have to know what is the difference between aerobic cellular respiration and anaerobic cellular respiration, and then one investigation. Let's just have a look here quickly. Now, when you look at the equation, I'll just do this so that you can see. Let me just get that other paper. There's the equation for photosynthesis. Here's the equation for cellular respiration. So it's just swapped around. The requirements for photosynthesis will now be the products for cellular respiration. So it's actually studying the same thing, like clicks 2 for the price of 1. So you can actually study this easier if you know how they are related to one another. Okay, you don't have to be able to balance the equation. You don't even have to know the chemical reaction. You just need to know the words. But in case your teacher handled it this way, you will know what it's all about. Okay, so please remember photosynthesis. Here we have cellular respiration. Okay, short summary just to show you the difference between the two. Cellular respiration happens in the cell. There's the mitochondrion, the powerhouse of the cell. It gives you the energy, and energy is carried in the form of ATP, adenosine triphosphate. That refers to the chemical structure. You don't have to know that. You just need to know ATP, energy carrier, and the energy we get from the process of cellular respiration. Here's the plant cell and the leaf, and there we have a picture of the chloroplast. That's the mitochondria, and this is the chloroplast. So how they interact with one another to help us to get photosynthesis and cellular respiration going. Now they talk there about aerobic and anaerobic cellular respiration. Now aerobic, in case you don't know how to remember this word, when you do aerobics, when you do exercise, you need oxygen. So aerobic oxygen is necessary, like they say there. When it's anaerobic, that word just shows us, that prefix shows us that oxygen is not necessary for this process to happen. Very important, you have to study this table. You must be able to write it yourself. So aerobic happens inside the mitochondria. Anaerobic happens in the cytoplasm, which is around the mitochondria. With aerobic, you need large amounts of energy to be made. When it's anaerobic, it is just small amounts. Okay? Aerobic, a cellular respiration, there's a total breakdown of the glucose, and with anaerobic, there's only partial breakdown. And then the end products, carbon dioxide and water. The end products here, carbon dioxide, lactic acid, and alcohol. So the lactic acid, that's also the lactic acid that you get in your muscles after some exercise. That is what's making you hurt when you feel stiff and you can't touch your toes anymore. And then the alcohol we also use when we make beer, etc. So this anaerobic process we use to our advantage. All right. It is also important to understand this process when you're interested in sport or athletics, or when you exercise. The science of sport, this will become very important if that is your thing. You need to know about that. All right. Again, just to show you, I have a few things that's important here. These blocks, they come from the ATPs. It tells us exactly. Aerobic respiration in cytoplasm and mitochondria use work and symbols like glycolysis, Krebs cycle, and oxidative phosphorylation. You need to know those three parts. Not the detail, no biological detail is required. Anaerobic respiration, production of lactic acid in muscles during exercise, the word and symbols, no biochemical detail of the process is required, and then the role of anaerobic respiration in the industry, like for instance beer brewing and bread making. Okay, here's just a picture to show you which type of activities are considered to be aerobic exercise, like they say cardio machines, spinning, running, swimming, walking, aerobic classes, dancing, cross country, all of those things. Anaerobic exercise are more like heavy weightlifting, sprints, jumping rope, hill climbing, interval training, and so forth. So that is why we will talk a little bit later about oxygen debt. When you do exercise and you run out of oxygen, you are tired, you have to take deep breaths, so your respiration rate will increase and the depth of your breathing will increase because the CO2 builds up in your body. Okay, so here is what is important. You have to compare aerobic and anaerobic respiration. You need one investigation, and that all depends on what your teacher did. And then they say CO2 is produced by living organisms for respiration. It comes from the ATP. And here on the right-hand side, you see there's a few words that I made a summary of. And I just want to talk about that a little bit quickly. When we now talk about the investigation, that is scientific skills. So every time we have an experiment, these are the things that they can ask you about. What is the aim of the experiment? Now, when we talk about aim, you look always for the sentence where they say to investigate. That's the one term that they use. I see they sometimes say to demonstrate is another indication. To investigate, to demonstrate. Then you know that will be the investigation. Okay, now the investigative question is asking a question. How do we ask a question? We're using a question mark. Many times I see when you write this investigative question, it's 100% correct, but there's no question mark. Then it's not a question. Okay. The other one, the hypothesis. The hypothesis must include two sections. For instance, let's say that light will influence the rate of photosynthesis. That's our to investigate if light will influence the rate of photosynthesis. Then when I look at the hypothesis, I will have two sections, A section and a B section. So the one will say less light will decrease the rate, or more light will increase the rate. Or you can just say it the other way around as well, increase in light will decrease the rate of photosynthesis, because the hypothesis, that is what you are testing. So it's not necessarily what you're going to see in your experiment. So there's more than one option for your hypothesis. Okay, so those three things, you can get that from this section. If they write this long experiment in your question and they talk about the experiment, then you go and look for your aim, your investigative question, your hypothesis, where they talk about to investigate, to demonstrate, for instance. They use words like that. To determine, that is the other one. To determine if. So look for the tips, and then you can identify them. Then the dependent variable, the independent variable, and a control. Always, we ask you about that. When you draw a graph, there's your heading, there's the y-axis, the x-axis. This will be the independent variable, that will be the dependent variable. Go and study that. In your heading, you must mention the independent variable and a dependent variable in order to get that mark. Don't be lazy. Don't want discount or summarize the heading. Like I said, then we summarize your marks. It's plain and simple like that. Independent variable is the variable that I can control. Just to remind you that it's the one that starts with I. So things like I decide the time frame. Am I going to check every 10 minutes, seconds, every few days, years, or whatever? So most of the time will be on the x-axis. The experiment that I used as an example, light influencing photosynthesis, obviously then the amount of light or the intensity of the light will be here, and that is my result of the experiment. I want to test the rate of photosynthesis, so that will be our dependent variable. That's the concept that I am testing. That's the result of my experiment. That will be here. And please, when you draw the graphs, don't forget the units. The units must be there. Then two other very important things that can confuse you. What makes this experiment valid and what makes this experiment reliable? Okay, so the first one, validity, means that what must stay the same in the experiment? The same type of plant, the same amount of water, the same conditions if you are not testing conditions. Everything that must stay the same makes this experiment valid. Okay, reliability, two things that you need to study. What makes an experiment reliable? When you repeat the experiment several times and when you increase the sample size. Okay, so that you study. Increase the sample size. Nowadays, we change this around. We don't just ask you, what makes this experiment reliable? We can actually say, why did we repeat the experiment several times? What is the reason that we repeated the experiment several times? Then you say, to improve the reliability of the experiment. Or we can say, instead of using 5 samples, we are using 50 samples. Why are we doing that? And you will say, to increase the reliability of the experiment. Okay, so those things, the skills that you need to test, please make sure it will always be grade 10, grade 11, grade 12. It's always in your exam paper. Okay? Next one. Effect of exercise. I just want to remind you about this again. We can take the pulse there. We can take your pulse right there. Here's a nice graph that shows you what happens before exercise, while you're exercising, and after exercise. And this is the pulse rate. So the pulse rate of a person that is fit will normally be lower than an unfit person. And something that you need to see, when you are unfit, it takes a longer time to recover than when you are fit. You recover quicker when you are a fit person. That is why when you have a tournament, you sometimes struggle when you're playing lots of games close to one another because you haven't recovered yet, and then you have to obviously play the next game. Very important when it comes to the breathing, interesting facts. In a sprint, you cannot breathe fast enough. So respiration takes place without oxygen. You remember previously we said sprinting is anaerobic respiration. So energy is still released, but much less. Lactic acid is made, lactic acid that's causing those cramps and stiffness in your muscles. Here they say this is poisonous and causes the cramps. Oxygen needs to be paid back to remove the lactic acid, and this is called oxygen debt. Watch sprinters after a race gasping for air to repay the debt. So the moment your body realizes that you don't have enough oxygen or you have too much CO2, it starts to increase the rate of respiration and the depth, how deep you breathe. Ne? Okay, so keep that in mind. Those are the things that we like to ask. And there it is, the first question. That is very important for today. We are going to look a little bit at the work and then do the questions about the work. This comes now from old exam papers, so let's have a look. How do we approach this? They say, the diagram below shows the leaf in a de-starched plant. De-starch, meaning you left the plant in a cupboard for a while so that it can use up the starch that is available so that you can do your experiment. Okay? It was set up by a learner to perform an experiment on photosynthesis. Study the diagram and answer the questions below. Okay, here's the first one. What was the aim of this experiment? Now, unfortunately, here there's no information. There's no to investigate, to demonstrate, to determine. So you have to use your brain in this one. So this is about what is happening when this plant is destarched. So we have to look at photosynthesis. So we want to know if we close this up with a piece of paper, if we close this leaf section up, that area will not receive any light. So this is now about light. Okay, so what is the aim of the experiment? It's about light and photosynthesis. So to demonstrate that light is necessary for photosynthesis. There's the two sections, light and photosynthesis. If you use other words to demonstrate that light is needed for photosynthesis, but that's the two things that's important. All right, next question. What was the dependent variable in this experiment? Now, if you don't know immediately, I always say draw a graph. See if you can draw a graph for yourself. The independent variable will be here. The dependent variable will be there. So now I want to see if light energy is necessary for photosynthesis. So what am I going to do? I'm going to change the light intensity, the amount of light. So here will be less light, there will be more light. So light will be there because I can control this. I decide the amount of light because I want to see what is the result, what is the rate of photosynthesis. Okay, so then maybe it helps you to see that this is about the presence and absence of starch. How will we know that? The rate of photosynthesis will be high because of the starch that is there, and the starch and the glucose will most now, when you remember the equation, we have CO2 plus H2O, that will give us C6H12O6 plus oxygen. So that is the starch, and that helps us to have photosynthesis. So the more we have of this and the more we have of starch, then obviously that will help in this process. This one and this one, CO2 and H2O, that's all of the things that's going to influence the rate of the photosynthesis. Okay, so the dependent variable, if I have this experiment, light, energy, and photosynthesis, light will be the independent variable, the rate of photosynthesis will be the dependent variable, but because they're here talking about the destarched plant, that is why they want the presence or the absence of starch, because if there's no starch, obviously this experiment will not work. What chemical was used to test for the presence of starch in this leaf? That you need to study. What reagent do we use? Here it is, iodine. We use iodine to determine if there's starch. And now they ask you, what is the color change? In other words, when it's present or when it's not present. Okay, so when it's exposed to light, obviously that is going to be made. Starch is going to be made, or sugar is going to be made in the process of photosynthesis. So when it's present, when it's there, it turns blue-black. Okay, if it's not there, it remains brown, or they can even say there's no color change. And that is the trick in this question. What was the color change observed in the areas of the leaf that were not exposed to sunlight? So if there's no sunlight, there is no photosynthesis taking place, so it will not change. It will remain the same because no starch will be made. If it's exposed to the sunlight, sunlight is most needed for photosynthesis, remember, sunlight, then there will be starch, and then it tests positive for starch. The iodine, it will turn blue-black. Okay, so just go and have a look. Do you see how they start? They start basic, and then the application of the knowledge that you have. All right, that was now photosynthesis. Let's look at the next one. Here we have an experiment. You will have this in your book as well. An experiment was conducted to investigate the effect of light intensity. What do they want to do? They want to investigate the effect of light intensity on the rate of photosynthesis. There's the aim, to investigate. The apparatus was set up as shown in the diagram, and the results are represented in a table below. Here we have a lamp that's going to give us light. There we have a test tube upside down, and then we see the bubbles there, the bubbles that will be forming, the water plant we have there. And obviously it's a water plant because that plant can function underneath water and photosynthesize underneath water. And then it forms CO2, and CO2 is the bubbles of gas that we see there. And the water has sodium bicarbonate. You will see that now. What's the purpose of the sodium bicarbonate? I will not give it away. Then we have a table. After this experiment was done, there's the ruler. The ruler is to show us that's the distance that the lamp, the light, the intensity from the experiment. Obviously, the closer we take the lamp towards the plant, the higher the light intensity, and then, as we know, the better photosynthesis. Okay, so here we have 3 columns. Distance between the plant and the light source in meters. Okay, 1 meter, 0,5, 0,25, and then 0,125, meaning far away to closer. And there's the light intensity that we have. When it's far away, the light intensity is less. When it's closer, the light intensity is higher. The number of bubbles given off in 1 minute. So when the lamp is far away, there's bubbles, but not a lot. When the lamp is close, there's lots of bubbles. Okay, so you need to understand the data. Then, here's the question. State a reason for choosing a water plant instead of a terrestrial plant in this experiment. What is terrestrial? Terra is earth, so a normal plant that grows in the ground. Okay, so why are we doing this? There's our answer. Water plants are structurally adapted to perform photosynthesis underwater. So it has a specific structure for its specific function. Okay. This one can actually count 2 marks as well. How is the plant adapted? All right. Now the next question that we have there, the addition of sodium bicarbonate. Sodium bicarbonate is the normal baking powder that you use. Why do we have the sodium bicarbonate? Because it is actually helping this photosynthesis process. An addition of small amounts of sodium bicarbonate increases the concentration of CO2, and it brings about a steady rate of photosynthesis. Okay? Next question. How was the rate of photosynthesis measured in this experiment? There we have it, by counting the number of bubbles released. Here we have the number of bubbles. Okay, that is our result. Then they say, with reference to the results given above, what deduction can be made with regard to the relationship between the intensity of light and the rate of photosynthesis? Now, when I look at this, I see the closer the light gets to the plant, the more bubbles is formed. So in other words, the relationship that we have here is, as the intensity of the light increases, the rate of photosynthesis increases. So it's directly proportionate to one another. Okay. Then what is important here, a further increase in the light intensity. When it's very, very close, the rate of photosynthesis will become stable at one stage. Okay. Then the last question here, tabulate 3 differences between photosynthesis and cellular respiration. Okay, when we have the word 3 or 2 or 4 or 5 or whatever in capital letters, it means I can only mark, as the teacher, I can only mark the first 3 things. Here it's very small, but it's just to use an example. There's photosynthesis, there's cellular respiration. I have a table. Please close your table for 1 mark. And then there's different things that you can use. But I'm only marking the first 3. So photosynthesis takes place in the chloroplast, where cellular respiration takes place in the mitochondrion. Photosynthesis needs light, and then cellular respiration can take place in the absence of light. Photosynthesis, it's anaerobic reaction, and then cellular respiration is a catabolic reaction. When you have science, you know what that means. Photosynthesis produces glucose and oxygen. Cellular respiration produces energy and carbon dioxide. So any four of them. Again, you can see how we can use the theory and we can ask you application questions. Very important that you practice questions, ladies and gents. I see it many times. You study the work, you know your work, but the way that they ask it confuses you or confuse you. So you have to practice this. There's old exam papers, use them. Practice, look at how we want the answers to be written afterwards. Okay. Next topic, gas exchange. So what do you need to know about gas exchange? Let's just look at this again. Gas exchange, very important, carbon dioxide control. You'll see carbon dioxide, homeostasis. It's grade 12 work, so we like to focus on that because you're going to do it again next year. Just quickly, when we go to the ATPs again, all of the ATPs, if you don't know what you need to study, you look in the ATPs and they will tell you exactly what is important for gas exchange. Let's just get there. Human gas exchange. Here they say the requirements for efficient gas exchange, all those different parts, how they must be adapted. Okay, they ask you what's the difference between cellular respiration, breathing and gas exchange. You have to know all the different parts. You need to know how ventilation takes place. And here's the one I'm talking about, homeostatic control of breathing, CO2 and homeostasis. Very important, you need to know that. Okay, so let's have a look just quickly. What is breathing? What is cellular respiration? What is gas exchange? Have a look. And remember that you must be able to tell me if I say differentiate between breathing, cellular respiration and gas exchange, you have to tell me what it is and how it is different from one another. Okay. Another picture, I get this from Mindstream. It's a very nice summary. It gives you the different parts of the lungs like we have here. And then every time they give you where it is, how it is built, and then obviously what is the function. This is a nice summary that you can use to study. Make sure you will be able to identify the different parts. Identify what is the structure like and what is the function. Make sure you have all those different things covered. Requirements for efficient gas exchange. This comes from the ATPs. So that's the different things that's important. Large surface area. It must be thin. It must be moist, well ventilated, protected, and what is the transport system that it has. So go and study that. Okay, here's a summary just to show you again. What's the requirements for efficient gas exchange? That is what you must have. Study, 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 ladies and gents. Okay. Homeostatic control of breathing. This picture is nice and colorful. It gives you the heart and the lungs there. So I just want you to focus on this. Normal CO2 levels here, when you don't have too much or too little CO2 in your body. Now what happens to change the CO2 levels? You can have exercise, physical activity increases. When the physical activity increases, you need more energy. To get more energy, cellular respiration takes place. Now when cellular respiration takes place, you're using glucose, you're using oxygen, and it forms CO2. So the amount of CO2 that is produced, it's increasing. Now, in your brain, the medulla oblongata is stimulated because it receives the message. There's an impulse sent. Receive the message that the CO2 concentration increased, and that's not good. CO2 can be toxic. So the impulses are sent, one to your heart and another message to your lungs, because your heart must now beat faster. When your heart beats faster, your blood pumps quicker, so the veins can pump the oxygen-rich blood quicker to the heart so that you can breathe it out. What is happening with the breathing rate? You are breathing faster, like that when you're practicing, and you are breathing deeper to get more of this out. And this is what it is showing you. The heart rate increase, blood with CO2 brought faster to the lungs. The CO2 is breathed out and the CO2 level drops back to normal. With the breathing rate increases and the depth of breathing increases, which is not mentioned here, and then the CO2 is exhaled. So this is very important for now, and this is very important for grade 12. Make sure you study this. Okay, another mind map. This one is derived of pictures and color. Maybe you don't study like that. So this is to show us normal CO2 levels in the blood, CO2 increase, and you can study this word for word. The receptor cells in the carotid artery are stimulated and it sends impulses to the medulla oblongata. The medulla oblongata stimulates the breathing muscles and the heart. The depth and the rate of the breathing increased. More CO2 is exhaled and the CO2 levels go back to normal. Those words that I'm using, very important. Please make sure that you study that. Okay, just something else I want to mention, exercise and altitude. Okay, sometimes athletes train at higher altitude levels because it temporarily gives them a benefit. The red blood cells carry the oxygen on a molecule called hemoglobin, and people who train at higher altitudes, they make more red blood cells. Okay, so when they come back to sea level, they actually get more oxygen, they can make more energy, they can run faster, and then after a while, obviously, the benefits are lost. It only lasts for 2 to 3 weeks. Okay, and another thing when it comes to pressure, lower altitude, greater pressure, more oxygen, higher altitude, lower pressure, less oxygen. Just go and have a look that you understand that. And this is the next topic, excretion in humans. All right, we go back again to our resources. So excretion in humans, NB, water and salt control, 12 marks in the test, 32 in your paper. So yes, there's lots of stuff to study here. Consult your ATPs and see what your teacher is asking you to study. But excretion, this is excretion in humans. In the skin, it's sweat and salt. In your digestive system, it's feces. And then kidney and bladder, urine, lungs, oxygen and CO2, and the liver give off the urea, which is a waste substance. Okay, excretion, know the different parts. You must be able to label it, know their function, know the terms that we have here, ingestion, digestion, absorption, assimilation, egestion. Life sciences, terminology, very important. Okay. Another thing that's important here, the enzymes that plays a role. We like to ask that as well. So in the mouth, in the stomach, in the small intestine, you see in the small intestine we get quite a lot. Carbohydrates, proteases, lipases, they have there, and there's the functions. So always when you make summaries for yourself as well, put it together, then at least you don't have 10 papers to study. It's just one table. Summarize it in the table for yourself and then study that so that you know when you are asked in the exam. Okay, so let's look at the first one we have here. There's the naked boyfriend. Okay, you can, before you look at the questions, you can actually label all of the different parts here. We have the esophagus, we have the stomach, we have the liver, we have the pancreas, the gallbladder, large intestine, small intestine, and then the anus that we have here at the end. Now they ask you here, write only the letter. Sometimes they ask you the letter and the name. Just make sure. Okay, write only the letter of the part that stores bile. Okay, and here we go. G would be, the gallbladder would be the one that stores bile. So if you study this to label it, when you do the summary like this, add the functions there. So you label them and you add what is the function, so that when you get a question like this, you will be able to answer this. Where will we find most of the enzymes? Obviously in D, there's D. We just saw it on the previous picture. Okay, let me just show you again in case you forgot. Here it is. Small intestine. Look at all the enzymes that we get there. Okay. Then the next one, where excess carbohydrates are stored in the form of glycogen, and that would be C. No, I'm lying. It's C. We have the insulin and the glucagon. I'm now confusing you here. That is where we have insulin and glucagon. They are secreted by the pancreas. And then where there's excess glucose, the glucose is stored in the form of glycogen in the liver here. And the moment you need it again, actually not just in the liver, also in the muscles, the moment you need it again, then the glycogen is converted back to glucose, it goes back into your body, and you can use it again. Don't make the same mistake as I did. The moment I think about insulin and glucagon and sugar, I think pancreas. Excess carbohydrates stored in the liver. Okay, where do we find most water and mineral salts? Where are most of them absorbed? Obviously in F. Okay, so when you do the summary, I say again, use a picture, label it, what is the function, because that's how we like to ask you these questions. Okay, next question. Human nutrition, we're still busy with that one. Instead of giving you a picture, now we just use words. Okay, the diagram below shows a diagrammatic representation in the digestive system. 2.3.1, name the part of the alimentary canal represented by X. So here we see there's the mouth, there's the stomach, small intestine, large intestine. Now there's our picture again. There's the mouth, there's the stomach. What is in between then? Obviously, the esophagus. Okay, here we have that. And ladies and gents, esophagus, many times you struggle to spell this, so just write it out several times. How do you practice for soccer to be good? How do you practice to kick a goal? You don't do it once, you do it many times, and that is how you will be able to spell this word. Write it out many times. Okay. 2.3.2, by which process is food moving through this structure? By, and I can do this, I don't know if you can see that, peristalsis. When you think about that esophagus, the muscles, they press it down through the movement of peristalsis. Okay. Then 2.3.3, name the glands P and Q that release their secretions into the small intestine. Now, doesn't matter which way around, it can be P and Q or Q and P. And then here we have it, the liver and the pancreas, or the pancreas and the liver. That is the answer we want. And then explain the chemical digestion of proteins. Let's look at the words that we're looking for. There's glands in the wall of the stomach, and it secretes gastric juices. And the gastric juices contains enzyme, and that enzyme is called proteases, because proteases works on proteins, and that helps to digest the protein to form polypeptides, peptodes, and then we have the building blocks that it becomes eventually amino acids. Okay, protease functions at an optimal temperature and in an acidic pH. Okay. So there you have it, and you must know in the stomach is very acidic. So different ways to ask the same type of questions. Okay, this one is also about the digestive system, the villi, villus. When it's just one, you find this in your digestive system, and it's built like this to increase the surface area. So let's look at the question. The diagram below illustrates the microscopic structure of a villus. Study the diagram and answer the following questions. So you go and you have a look. You must be able to label. When I look at this, for instance, 5 looks like the columnar epithelial cells. 4 looks a little bit different. You see it looks like a bucket. That's the goblet cells. 3 is the section here inside, which is the lymph vessel, and this one we call the lacteal. Number 2 shows the sides, which will be the capillaries. That's all around. 1 and 2 actually, it's part of the capillaries. Okay, now give the function of the part labeled 3. Now 3 we said is the lymph vessel. Okay, so if you don't know what it is, you can't give the function. And the function is absorption and transport of fatty acids and glycerol. Now fatty acids and glycerol is nothing else than the building blocks of fats and oils, or the other word that we use, lipids. 2.2.2, which labeled part contains blood with relatively a higher amount of glucose and amino acids? Now the blood parts would be number 1 and number 2. Ne? Now 2.2.2 is labeled one part, labeled 1. That one will have all the glucose and the amino acids in, that's being absorbed, that's now going to the body. All right. 2.3.3, name the process that enables humans to absorb the nutrients. That would be diffusion, active transport. Then lastly, 2.4.4, explain how the villus is structurally adapted to enhance the absorption. In other words, how is it built to do its job better, to do the absorption and the digestion of nutrients? And here we go. It's only 1 cell thick. It's thin. You see, there's just 1 layer, so the nutrients can pass through quickly and easily. That's the structure and that's the function. That's how you answer. The next thing, it's richly supplied with mitochondria. What is mitochondria giving us? Energy. So it supplies energy for the active transport. It also secretes mucus that serves as a carrier fluid for nutrients. They're in the goblet cells. Okay? The moist membrane enhances the diffusion. You also have a moist membrane in your lungs to help with the diffusion. It has microvilli, very, very, very small villi in your digestive tract like this. So what happens? It increases the surface area. Okay? And there you see 3 times 2. You look at the mark allocation. Many times you have to mention something and give the function. That would be section A, section B for 1. So it's always an A and a big section. You mention something and you explain. All right. Human nutrition. The next section that's very important. When we look at our guidance here, it says water and salt control. Like it did with carbon dioxide, it's now talking about water and salt. This is also part of the grade 12 syllabus, so I suggest you pay lots of attention here. Teachers are going to ask you this now because you're going to be in grade 12 next year. Just to recap, the nephron is the functional unit of the kidney. There you can see inside there, we get the nephron. This is how the nephron looks. You need to know the different parts: afferent, efferent, arterial, glomerulus, Bowman's capsule. Why is Bowman's capsule looking like this? Why is it built a specific way for its specific function? Here we have the proximal and the distal convoluted tubuli. Here we have the collecting tube. There's the loop of Henle. And then obviously, first capillary network, second capillary network, it is entangled because in the kidneys you need to reabsorb the nutrients, the water, the salt that is still needed so that you do not urinate it out and you lose it, or when you need more water needs to be reabsorbed. Okay, so here we have what we call negative feedback or homeostasis or controlling of the water levels. Here in the middle, normal water levels. So whatever happens, every time we need to come back to normal because we need balance in the body. Okay, so this you study. When the water levels decrease, when you dehydrate, when there's not enough water in your body, what is happening? Okay, the pituitary gland secretes more ADH. So there's a gland involved. That is very important. And there's a hormone involved. That is very important. Okay, so the gland is the pituitary gland. The hormone is ADH, which is antidiuretic hormone. We're going to ask you that. And then what happens when the pituitary gland secretes ADH? The permeability of the collecting ducts and the distal convoluted tubules increase. In other words, more, diffusion can take place, more absorption can take place, reabsorption, so that the permeability of this wall is now better. Okay, more water is reabsorbed into the blood. So what happens if there's more water reabsorbed? Okay, there will be less water in the urine, decrease water loss. So the water that goes out in the urine will be less because you want more water back in your body, and that gives us concentrated urine. Remember that. Now the other way around as well, when the water levels increase, the pituitary gland stops secreting ADH, or less ADH is secreted. The permeability of the collecting ducts and the distal convoluted tubules decrease. We don't want a lot of water to be reabsorbed. We want it to be urinated out. So less water is reabsorbed, more water is lost, and that will give us dilute urine, and the water levels go back to normal. NB, study this. Okay, the same with salt. There we have the nephron, which is the functional unit of the kidney. There's pictures that is explaining exactly the same as this, homeostasis or negative feedback. All right, there's the kidney, and I always say, where will you find these hormones? It looks like a little cap that's on top of the kidney, and that is going to secrete, the adrenal gland is going to secrete aldosterone and adrenaline. In this case, where we talk about salt, where the salt levels decrease, the gland is going to be the adrenal gland, and the hormone is going to be aldosterone. It causes reabsorption of sodium. Sodium is salt. There it is. And then the salt level increase. The same this way around, the salt level decrease. We have the same gland, we have the same hormone involved, reabsorption of the sodium, and then the salt levels go back to normal. Please study that. Okay. Glucose, also important for grade 12. The same situation. Homeostatic control, negative feedback. Here's more pictures involved for you. All right. This side is just to show you, this is the pancreas. In the pancreas, we have the islets of Langerhans. There we have alpha and beta cells, and these cells secrete the hormones that's responsible for controlling the glucose levels in the blood. The one is called insulin, the one is called glucagon. When the blood glucose levels go too high, insulin is secreted. When the blood glucose levels are low, glucagon is secreted. And I always use this to help you remember. When the glucose are gone, we need the hormone glucagon. Okay, and where do we get the glucose from? The glucose is stored in the liver in the form of glycogen. So then when we don't have enough glucose, this is the glycogen is turned back into glucose. So look at these words, ladies and gents. Glucose, glucagon, glycogen, they confuse you. Again, you have to write this down several times. We know it confuses you, so we ask it. And it is grade 12 work. It is very important. Okay, so here we see homeostasis, normal blood glucose level. When there's an imbalance, when there's a increase in blood glucose levels, the pancreas secretes insulin. Okay, it stimulates that more glucose is taken up, and the excess glucose is stored in the form of glycogen in the liver, and then the blood glucose level return to normal. The same can happen when there's not enough glucose. We short glucose, the glucose are gone. The pancreas will stimulate glucagon. Glucagon will make the glycogen turn back or break it down back to glucose, and then the blood glucose levels will increase back to normal. Please go and study this. All right. Questions. Here we go. The table below represents the concentration of substances in the blood of the plasma, glomerular filtrate, and the urine. There they show you. That's the plasma, the filtrate, and the urine. So water, protein, glucose, sodium, and urea, waste product urea. Okay. 3.2.1. Which substance that was present in the blood plasma did not filtrate through the Bowman's capsule? And give a reason for this answer. There I see it's in the plasma, but it's not in the urine, so it will be protein. And if you remember your work, you will know that protein molecules are too big. They can't go through, okay? It is protein and it's too big to filtrate. Explain the following. No glucose in the urine. When I look at glucose, I see no glucose in the urine. Obviously, it's being reabsorbed by your body. Glucose are actively reabsorbed in the proximal convoluted tubuli because glucose is important. It gives energy to the body. Look at the mark allocation. You have to elaborate if the mark allocation is more than just 1 or 2. A higher concentration of urea in the urine, it's a waste product. Urea is a metabolic waste product. It is excreted from the capillary veins to the nephron in the distal convoluted tubuli or tubule, so 3 marks. Now, 3.2.3. Does the person with the medical report show above that he suffers with diabetes mellitus? In other words, diabetes. Now, this person will not have diabetes because when you have diabetes, either the insulin is not produced or the amount of insulin that is produced is not sufficient, so glucose cannot be stored or used, so there will be glucose in the urine if you have diabetes. Okay, what do they say? If a person suffers from diabetes, there would be a certain percentage of glucose in the content of structure B. A diabetes sufferer will have an abnormal glucose content in the blood because his insulin secretion is abnormal, either not at all or not sufficient. Therefore, glucose is not converted to glycogen, hence the glucose is excreted through the urine. Okay, there we have no, that's the compulsory mark, and then 3 other things. Okay. The next topic, biodiversity. Again, you have to make a summary for yourself. This is the mind map that I get from Mindstream. What do they say about biodiversity? Okay, we look at this, biodiversity and classification of microorganisms, the role of bacteria in biotechnology. 29 marks or 10. So here we have the different classes, groups, bacteria, protista, fungi, plants, and animals. You need to know what's the role. You need to know effects and management of diseases. You need to know about these things. And then I included this because vaccination is a big talk about that currently with COVID. So maybe you will get a question about that in your exam paper or in your test. So make sure you understand what is happening there. Okay, biodiversity of plants. I have pictures here for you as well. That helps you to understand that. Here obviously we have the gymnosperms. There we have the angiosperms. Here we have the pteridophytes, which is the ferns. And there we have the moss plants, which is the bryophytes. Okay, so we're always going to ask you questions where you compare the angiosperms and the gymnosperms, the pteridophytes and the bryophytes. So please make sure when you study them, here's the mind map, bryophytes, okay, pteridophytes, angiosperms, gymnosperms. There they talk about asexual and sexual reproduction. Go and have a look how the flower is built for pollination. Okay, these are the questions that we can ask you. We can ask about the seeds and the fruits, or the cones rather, that we have in the gymnosperms, where there's sometimes seeds and no spores, where we have xylem and phloem, conducting tissue, any of those things. You have to compare them. Is water necessary for fertilization, etc.? Go and have a look at all of those things. Make a summary for yourself like this, and then you study. Look at the things that's important. Is it a talus plant or not? What is a talus plant when you don't have true root stems or leaves? What is vascular tissue when you have xylem and phloem? Do they use seeds and or spores? Will there be fruits? What type of reproduction? What's the dominant stage that we have here? Gametophyte or sporophyte? Examples of them, and then the image that's unfortunately not colorful, but this is how you summarize it for yourself and you study this, then you can answer the questions. That's the plants. Okay, so let's have a look. The diagram below represents the different phyla of plants. Okay, there they show you this is a moss plant, this is a fern plant, a pine, and a flowering plant. Okay, now identify the plant groups. They want the group name, A, B, C, and D. So A will be the bryophytes, fern will be the pteridophytes, pine will be the gymnosperm, and then the flowering plant will be the angiosperm. So have a look. When they ask you plant groups, you must give the plant group name. Okay? Explaining by referring to reproduction why plant C is better adapted to survive on land. Okay, angiosperm and plant A is the moss plant. So when we look at this picture, the moss plant, it's a very, very, very small plant. And then the gymnosperm, it can be tall trees. So now they're comparing these two. They ask you about this. Okay, so here we go. Plant C has vascular tissue, xylem and phloem, and does not depend on water for reproduction. Plant A does not have vascular tissue and depend on water for reproduction. 2 times 2. Tabulate 2 differences between gymnosperm and angiosperm. There we have it. Okay, naked seeds, seeds enclosed in the ovary or a fruit. No fruits have fruits. So if you study that table, you will be able to answer this. The flower in plant D is a sunflower. Other than its bright color, justify why this flower is suited for insect pollination. Look at how it is built. And here we go. The anther and the stigma is on the outside of the flower. Okay. Look at these questions. Make sure you will be able to answer them. And now we look at the flower, different parts of the flower. Don't forget. Try to label this before you start. Okay. What do we call that one? That one would be the anther and the filament, because that is the male plant. Anther and filament. That is the male part of the plant. That is the male part. This is the female part. So there we have the stigma, the style, and the ovary. Then we have the colorful leaves here, and then we have the sepal, the green leaves here at the bottom. So these leaves are colorful to attract pollinators. So now here they say, give the letter and the name. So the letter and the name. Don't, skip that and forget about it and just write the letter or just the name. So which part here produces the pollen? Here we have it. A, the anther. So that one. I'll talk about this now. I'll show you. Which part receives the pollen? Which will be F, the stigma. And then which part attracts the pollinators? Which will be the brightly colored leaves here, which is C, the petals. Now, let's say for instance you have anther right, but you have the letter wrong. Okay, then you can still get 1 mark. It is not linked to one another. If A is wrong, that one is also wrong. So you can still get the marks. All right, so that is biodiversity of the plants. Now, biodiversity when it comes to animals. Now have a look. You see symmetry and cephalization, coulomb and blood systems, tissue layers, and numbers of opening in the gut. Okay, and when you look at the key features that's important, those are the things that you need to know. What is symmetry? What is cephalization? What is a coulomb? What is blood systems and the different types of tissue layers? So you need to summarize this for the different sections. Here we have them. We have Porifera, we have Cnidaria, Platylamentis, Annelida, Arthropoda, and Chordata. Okay, examples of the different parts. And then very, very important, you have to have a summary of this again. That is how you study this, all the different ones. Porifera, Cnidaria, Platyhelminthes, flatworms, Annelida, the earthworm, Arthropoda, Chordata. And here I have mode of life. Symmetry, is it asymmetrical, meaning there's no symmetry? Radially symmetrical, circle, you see that? Bilaterally, by both sides. We have bilaterally symmetrical here, and then cephalization. Cephalization is when we have a head section, a brain section, eyes. Okay, cephalization, we don't have it in the Porifera and Cnidaria, but we have it in the flatworms. Okay, number of tissue layers. Diploblastic is 2 layers, di is 2. Triploblastic, 3 layers. Okay, presence or the absence of a coulomb. Acoulomate, meaning no, they don't have it. Okay, coulomate, meaning they have it. You also get a pseudo-coulomb when it's a fake one. Okay, presence and absence of blood systems, presence and absence of guts. All right. Here we have the words, terminology, and pictures to explain the different terms. Sessile, when you are sessile, you are attached, you can't move. Here's cephalization, the process of accumulation of sense organs at the anterior end, the front end. Here I have a lobster, they show you there. There's the eyes and the head, okay? So it's concentrated in the animal's head end. That is cephalization. Triploblastic, okay, diploblastic, triploblastic, it's the germ layers. There we have 3 layers. Here we have 2, mesoderm, endoderm, ectoderm. Okay, here we have the coulomb, acoulomate, pseudocoulomate, and coulomate. This one, there's no coulomb. Here we have one that is not real, and there we have coulomate has a body cavity, and this body cavity that does not completely surround the internal organs. Okay, so have a look there. This is symmetry, bilateral symmetrical, radial symmetrical, and spherical symmetrical. This one we don't do, we talk about those two, or asymmetrical when it's not symmetrical. So go and study the terminology, biodiversity. Okay, so here we have, let me just do this. Here we have questions. First we look at the diagram. I see the animal kingdom, the 6 phylums, Porifera, Cnidaria, Platyhelminthes, all the way to Chordata. And then they ask you, identify the phylum that is acoulomate. So now if you have that specific summary, this one that I talked about, you will be able to answer the questions. Okay, so is it acoulomate? That's the first one. Examples would be the Porifera, the Cnidaria, and the Platyhelminthes, okay? Who has exoskeletons? That would be the Arthropoda, okay? C has organisms that have segments. Which one will have segments? Like the earthworm and Annelida, okay? 1.5.2, name the type of symmetry that will be found in these phylums. The first one is Porifera. What will be the symmetry? It's asymmetrical. What is the symmetry in 2? 2 is Cnidaria. That will be radial, like the jellyfish that you saw. And then number 5. 5 is Arthropoda. They are bilateral, symmetrical. Number 6, Chordata, bilateral, symmetrical. So make the summary, study the summary, and then you will be sorted. Okay. Another topic that is very important, you will see here when we go back to our resource here. Biodiversity of animals, you must have a comparative table, and phylogenetic trees. We like to ask about that. Now, what is phylo? Phylo refers to phylum, phylogenetic trees. It's about the genetics. It is a branching diagram. There you can see examples. It's branching, and it's a diagram showing the evolutionary relationships among various biological species. It shows us differences, it shows us similarities when it comes to the genetics, and then very important, it can show us common ancestry. So this is also important for grade 12, so you need to understand, your teacher can ask you about the animal diversity or the plant diversity, and then you need to apply. You can see there they branch. every time when they branch. So these two are more related than, for instance, E, and C and D are more related than E. So make sure you understand how it works. There it shows you common ancestor for A and B. You will find there they are sister groups, and then C is the outgroup to A and B. They are not as related as taxon A and B will be. So just make sure you know. Okay. Population ecology, growth, population growth. What is important here? The factors that's influencing population size and the growth forms. Here's the two growth forms that we have. Geometric, the J curve. Logistic, the S curve. They talk about carrying capacity. Know what that is. Okay. Know the different stages of the curves, like the lag or the death phase or the geometric phase, so that you can answer the questions. All right, so let's have a look here. When we look at this curve, what is this? The J curve or the S curve? Obviously, this will now be the J curve. And what is the correct name for the S curve? That would be the logistic one. Okay, so there we have it, logistic. Identify phases P and Q. There's P and there is Q. So this is still in the beginning where they are still establishing themselves. So we call this the lag phase or the establishment phase. Okay. They ask you about Q. You can see it goes up. Now things are happening. This population is growing. So that would be the geometric phase. Geometric phase, also called the logarithmic phase or accelerating phase. Okay, give the letter only of the phase where the birth rate equals the death rate, where it's exactly the same. Obviously, ladies and gents, where is the graph stabilizing? It's not going up or going down, so they equal the birth and the death rate. Mortality and natality, that would be at S. And then where the birth rate exceeds the death rate by far, where is it increasing a lot? It's increasing a lot at Q. Okay. So, population ecology. Questions? Terminology that you need to know, I'm just showing you quickly. Immigration, emigration, mortality, natality, fluctuation, and carrying capacity. What is carrying capacity? What is the effect of all these different terms? Increasing numbers, decreasing numbers, and then what is carrying capacity? Go and study that. We like to ask you about those things. Okay. Now, a question about investigations. Here's the experiment. We did talk previously about the different things. Okay. I said you must be able to say what's an aim. You must be able to ask the investigative question, the hypothesis. Okay, that is important. What else is important? Dependent variable, independent variable, and what is a control? Also, reliability and validity. Okay, those are the things that we're going to ask you about. So let's look at this question. All right. Let's read the experiment. Learners wanted to investigate the effect of different antibiotics on the growth of E. coli. So what is the experiment? To investigate. That was my aim. They give you different steps. There they give you the results. Excuse me. So after 48 hours, they observed their results as follows. Okay, now, first I ask you what is the dependent variable. Now, if I draw a graph, dependent variable is there, independent is there. What am I investigating? The effect of different antibiotics on the growth. So growth will be there, and the different antibiotics will be there. Okay, so the dependent variable is the growth of the bacteria, like you can see there. What is the independent variable? Like we talked about, the different types of antibiotics. That's the independent variable. So always, if you're not really sure, see what happens when you draw a graph. That helps you. Then they say formulate an investigative question. Now there's our investigation. And you must ask a question. There's different questions. You can ask something like, which antibiotic will have the best effect on the growth of E. coli bacteria, for instance. Okay, which type of antibiotic is most successful in preventing bacterial growth? And remember the question mark. Okay, explain why no antibiotics was added there. That would be our control, so that we can compare the results. Have a look. Plate 4 is the control, and that is to compare the results. Okay. Then they say, how can the learners make this experiment more reliable? By repeating the experiment and increase the sample size. Okay. Then 2 precautions you will take during step 1. Step 1 is the preparation of the agar plate. What is important? Wear gloves, wear a face mask, use sterile equipment. The only question that they don't have here is how we will make this experiment valid. And then you go and you have a look. Same. What must you keep? Same. Each agar plate will have the same amount of E. coli bacteria and add the same amount of the different types of antibiotics. Okay, so be aware of that. Look out for that. You're going to have an investigation question, so make sure you are able to answer all those different questions that I talked about. Aim, question, hypothesis, dependent, independent, control, reliability, and validity. Okay, cover yourself. Now, ladies and gents, the last section, human impact on the environment. There's very nice summaries, mind maps like this, made by the subject advisor that you can use to study. Here it is about the atmosphere and climate change. Everything that you need to know is here. Go and have a look. Look at the ATPs. What do they say in our question resources? Focus on topics that were taught using the mind map. Okay, 37 marks or 15. And this is the mind map, and this is the topics. Okay, remember definitions. What is greenhouse effect? What is a greenhouse gas? What is enhanced greenhouse effect? What is global warming? You need to know that. What is your carbon footprint? As you can see, the next one, carbon footprint, everything that's important here. And then the ozone layer, they can ask you about that. And next one, the availability of water and the quality of water. All these different topics we can ask you about. Eutrophication, thermal pollution, mining, alien plants, what is the effect, destruction of wetlands, what is the effect of boreholes and aquifers. Please make sure you study this and that you know what's going on. That one is exactly the same. It's a copy. Okay, food security. What do you need to know about food security? Poor farming practices, which is monoculture, pest control, loss of topsoil, use of fertilizers, all of those different things. The mind map, study so that you can apply your knowledge. I'll show you now a few questions. What happens when we lose our biodiversity? Look at this. If we lose it through habitat destruction, poaching, alien plants. Okay, here we have deforestation, urbanization, Gulf estates, mining, loss of wetlands, and the alien plants. So make sure you know what is the effect of all of these things. Okay, solid waste disposal, like we have these landfills. What do we do? How do we recycle? How do we manage the dump sites? Very important. Okay, so here's the questions. Let's have a look how we ask you this. Long section to study. Now, when you get a case study like this, please read this properly, because every now and again you are lucky enough that they ask a question that comes out of that case study as it is. So use that. Now, the first one is, what is the process called when science are used to produce more food? Now, when you read here and you see what they did, they actually made burgers, using a lab-grown beef burger, because the fact that all the livestock is now contributing to the greenhouse gas emissions. So what do we call it when we genetically modify or change things? genetically modification or genetic engineering. Then they say name 2 ways in which this process can help combat food shortages. It's going to increase the yields, that will be the amount. It's going to increase the shelf life of the product. Shelf life, meaning it can stay on in the shop longer so that you can buy it and you can get it. Products is available throughout the year. Okay, then what is climate change? That is a definition. Any change in the long established climate patterns of a country or an area. Number D, according to the article, what is the percentage of global greenhouse gas emissions that is contributed by the meat industry? And there it is, the Christmas box, according to the article. So you have to read this, and when you read this, you will see it contributes about 18%. No study, just reading, there's a free mark. Okay, one thing that I want to add here, you need to know that when we work with cattle, rice paddies cattle, it is increasing the methane production, and that is a greenhouse gas. Just make sure you know all the greenhouse gases. Okay. And this one is about the same newspaper article, but it's more questions. They ask you to distinguish between greenhouse gas and the carbon footprint. What is a greenhouse gas? Gas is emitted into the atmosphere that contributes to the greenhouse effect. What is carbon footprint? We like to ask that. The amount of carbon dioxide produced by an individual's activities in a certain period of time. Then food shortage is a global problem. Define food insecurity. There we have a definition again. A state of being without reliable access to a sufficient quantity or affordable and healthy or nutritious food. Now what they did here is, normally they ask you what is the definition of food security. So now it's just the opposite of food security. So you use the definition for food security and you just say it is when you are without those different things. All right. Explain 3 threats to food security in the world. There we have it. The growth of the population, urbanization, floods and droughts, and food wastage. And obviously all of that causes increase in food prices. Okay. Suggest 1 practical solution to food insecurity. Learn to produce own crops, have lesser children, limit food wastage, use genetically modified seeds. So ladies and gents, these are the types of questions that you can expect. As you can see, with human impact on the environment, a lot of it is application. Okay. Now the last one, the last of the last here is again a graph. Okay, I just want to show you when we mark the graph, how we mark that so that you are aware. Here we have the heading, and in the heading sometimes we use an H, sometimes we use a C for caption. As you can see here, the heading counts 1 mark. You get 1 mark when you have both the variables. In other words, what is written here and what is written here must be mentioned in your heading in order to get 1 mark. The next one is T, the type of graph. In this case, they ask a bar graph. So if the type is correct, you get 1 mark. They say correct labels for X and Y axis plus the correct units. That is also 1 mark. So you have to do a lot to get the 1 mark. The name and the unit. The name, this is now the different types, and then you can get a mark. Appropriate scale, the spaces in between and the width of the bars. If that is correct, you get the mark for scale. And then the last one is the plotting. If you have 1 to 6 of them correct, you get 1 mark. And if you have all 9 correct, it is 2 marks. So that is how you need to make sure a graph can give you marks. You can get marks easily, or they can give you a graph and ask you to read information from the graph. So just make sure that you understand. Okay. In this same question, they ask about biodegradable and non-biodegradable waste. Obviously, food scraps is biodegradable. Wood will be biodegradable. Glass and metals will not be biodegradable. So if I can just get the camera back here to me. Ladies and gents, this is now in short what you need to prepare yourself for with the test, or if you write paper 1 or 2, practice, practice, practice. It's the last stretch. Good luck, look after yourself, try your best, and then if everything goes well, we will be seeing you again in grade 12.